gives the ACSR certificate. So under the ACSR umbrella, is also working as an assistant professor. So his current research activities include development of advanced hybrid nanomaterials for point-of-care diagnostics. He accomplished his PhD and MS in biomedical engineering, especially bio-nanotechnology specialization from the Department of Bio-Nanotechnology, Okan University, South Korea, under his Professor Hausi Yoon Group. During his research studies in South Korea, he developed a new class of functionalized metalloid, polymer, and carbon nanomaterials for antimicrobial and electrochemical biosensor studies. Prior to joining CSIR-CECRI, he was a DST Inspire faculty and PhD course coordinator at Nipar, Kolkata, West Bengal, from April 2016 to 2017. Dr. Murugan had the postdoctoral training from University of Montreal from April 2013 to February 2015, and from Quebec, professor under the Professor J. Su and Professor Jigyasa, and University of Guelph, March 2015 to March 2016, and from Ontario, Professor Nidhi Rajan, Canada. So vast experience in the postdoctoral research. During the research stay in Canada, he worked on 3-in-1 metal, dye, biopolymer, hybrid nanomaterial, and biofunctionalized graphene oxide system for biosensor. Until now, he contributed more than 55 peer-reviewed journals in international journals and 6 book chapters and 2 international patents to his credit. Now with this brief introduction, now I call upon Dr. Murugan Veerapandiyan. Sir, please.
Thank you, Dr. Angappanji, for the brief introduction on my background. It's my immense pleasure to be presenting my content for this Jigyasa workshop for teachers. I hope in the next 30 to 40 minutes, you will enjoy some of the content little bit far away from the scientific content of electrochemistry. So far you listened from others talk. So the title of today's talk is my slide is visible to you people? Can someone tell me, is that visible?
No, sir.
Yes, sir.
Yes, sir.
Yes, sir. Yes, sir.
Thank you. So, seeing the overlooked situations from the COVID-19 impact. I think from every day, morning and night, we are listening to this pandemic term, COVID-19, and histogram data on various statistical information related to infection, how the recovery number is declining, or new cases is recording in each and every part of the country as well as global. So there are plenty of situations or healthcare associated things which has been overlooked because of this hype came from various part. So the major part we have to look at our ecological system is healthy life at home, not necessarily at home, also in workplace. So if you maintain the healthcare system in your home, obviously your working environment will be healthy. So that is the fundamental concept. Staying physically active, looking after mental health, quitting intoxicants, healthy parenting, and eating healthy. I think this is the most important thing we have to follow, not necessarily in the pandemic situation, in the regular days. So whenever you follow healthy physical activity, mental health, and quitting of intoxicants, obviously you will lead a healthy life. Then you can apparently lead a parenting. When you lead parenting, you need lots of energy. So obviously you will eat healthy food. So how I imagined myself as teacher, I believe everybody in this presentation is teachers in reputed institution, government institution. So when you believe yourself as teacher, this is the expectations in the normal days. But the pandemic situations has made us a different look, right? Reality in online, always waiting whether the recipient is receiving my voice. Am I audible? Or the listeners are listening to my talk or to others? And always we have a doubt whether my content is reaching you or you are listening to your neighbors or somewhere else. That's the fundamental question always running in our head because of the online situations. And of course, working from home changed from time to time because of this pandemic situations. We have to pretend sometime and we have to really act differently at some point of time. So this is how happening everyday life in pandemic situations. So everything is accountable for our productivity, individual health as well as ecological health. So lockdown have made unprecedented situations in every individuals, not necessarily in one region, across the globe. So previous lockdown situation is like this, very busiest road system, busiest household activities, everything. But this lockdown situation have made serious unexpected things for not only the low income people as well as the high income people. The repeated term we listen from the televisions are isolation and quarantine. So there is a difference. I hope many of you might be familiar. Isolation of sick people or isolating you from sick people. That is one thing. And another thing is quarantine. Quarantine separates and restricts the movement of people who were exposed to contagious disease to see if they become sick. So these are the routine terminologies being followed by Ministry of Health for making a containment zone in particular regions. So let us have a look into virus structures. How this SARS-CoV-2 has been generated from their siblings. So in 1997, H5N1 came into our environment. You can see the structure of H5N1, and 2002 SARS, severe acute respiratory syndrome causing virus structure, is same kind of envelope protein as well as internal structures. In 2009, H1N1, this is like avian flu, and H1N1, the swine flu, which is having more or less same, but phenotypic and genotypic variations are little bit changed. In 2012, MERS came with a severe fatality. Compared to SARS, MERS is less contagious, but in opposite it is very, very fatal. And this is the 2020 SARS-CoV-2, which is also having a similar kind of phenotype with the earlier flu viruses. World Health Organization have validated different amount of diagnostic assay methods for confirming the existence of COVID-19. Among that, reverse transcription polymerase chain reaction, the so-called RT-PCR, is the confirmatory assay for confirming the existence of COVID-19. In particular, E-gene assay, RNA-dependent RNA polymerase gene assay, and N-gene assay. So in line with these, I will explain in the following slides what is meant by envelope assay, that is E meant for envelope gene assay, and RNA-dependent RNA polymerase gene assay is for amplifying the RNA of viral, and N means the internal nuclear protein associated gene amplification using RT-PCR. So these are the confirmatory assays validated by World Health Organization to confirm the existence of COVID-19. There are plenty of alternative assays still in R&D. Some are in commercial market for reliable applications. One among them is calorimetric based RT LAMP assay. LAMP is abbreviated for loop mediated isothermal amplification, which is also kind of amplification of nucleic acid related to pathogen, maybe of bacteria So this is also one of the promising assay technique employed for making the existence of or detecting the existence of SARS-CoV in suspected clinical samples. So E gene assay, as I mentioned, E means the envelope small membrane protein. So whenever a patient is having viral proteins from air or some contaminants, these spike proteins associated with SARS-CoV-2 virus or any virus will look for suitable receptor in our biological system. So with respect to SARS-CoV-2, angiotensin converting enzyme ACE2 act as the receptor to provide seating space for SARS-CoV-2 spike protein. So if we are able to amplify the envelope proteins associated RNAs using RT-PCR, then we can tell the existence of viral antigen. The next confirmatory assay is RNA-dependent RNA polymerase assay. This is also exactly measuring the internal RNA genome exist in the viral. And additional confirmatory assays are nucleocapsid genes of SARS-CoV-2 analysis. So this is how SARS-CoV-2 virus look like, and these are the variety of biorecognition factors which can be used for assaying the viral existence. Of course, this nasopharyngeal and oral pharyngeal swab collection is painful because of the contagiousness. People are preserving them at one location, and the victims or the suspected patients are keeping their distance, physical contact away from the clinicians or healthcare workers. And it is a really kind of annoying procedure, collection of this swab, and keeping them ourselves in the viral media so that better preservation of viral antigens can be done. So RT-PCR, how it really works. So this is how polymerase chain reaction works. Step 1 is denaturation. So unlike well-developed eukaryotic system, prokaryotic system will not have double-stranded RNA at some point. At some point it has coiled DNAs in bacteria. So in any case, the viral RNA should be broken down into single-stranded structure. Then it has to be complementarily bind with other nucleotides, or we call as RNA primers, and it has to be amplified into the database or pre-programmed sequences matched with the existing viral things. So this is how polymerase chain reaction works. So reverse transcriptase enzyme is used to re-amplify the RNA into DNA so that we can confirm the existence of viral associated RNA in the suspected clinical samples. So this is the work principle of PCR and RT-PCR. And there are plenty of recent bioassay techniques are evolving in the R&D system as well as commercial point of view. These are the similar kind of biosensor demonstrated for different other pathogenic infection detection. For example, lab-on-a-chip technology have evolved into a point of care diagnostic system for rapid detection of plenty of bioanalytes. You can see here there are different amount of inlets where one pathogen sample can be injected, and magnetic beads pre-modified with the antibodies or nucleic acid can be then allowed for binding or incubation at some point of time. So when you inject wash buffer, the unbound antibodies or antigens from the suspected pathogen sample can be washed off, and then it can be separated into different detector system. Here the chip based biosensor is demonstrated for loop amplified probe reagent, by which the specific amplification of nucleic acids lysed from the infection causing agents are amplified for biosensor studies. So this is another paper-based biosensor platform which is well known for various diagnostic procedure. This is how the pregnancy kit also working. Whenever urine sample from pregnant women is placed on here, the urine sample having human gonadotrophic hormone will bind into the suitable receptor here, and we will be following a test reading here in association with the control line. Same way, immunological assay for COVID or any other bioassays can be performed, provided we have to modify the antibodies related to IgG or IgM of particular infectious pathogens modified on this particular area. Then if the relevant antigen is coming from the patient sample, means it will bind to the antibody, by which you can confirm the existence of specific antigens in the victim's solutions or victim's biological fluid. So this is another paper-based biosensor technique demonstrated or projected for infectious diagnosis. Next one is film-based biosensor. Same way of optical imaging can be done. Apart from that, the same immunoassay also expressed in thread-based biosensor. Thread means nanowire or microwire kind of fibrous structures are done. Even right now, LG company have launched a pen on a chip. So a pen having an electrode chip, and when you unscrew, and you can just add your biological fluid on tip of the pen. When the pen containing antibody or other biorecognition element is getting complex with the analytes, it will capture the analyte, and those bonding will change the optical signal or electrical signal or electrochemical signal and display the abnormality in the readout system, by which you can tell the existence of antigen in biological fluid. And this is one of the classical example related to glucometer, which is actually based on 3 electrode system, like working electrode here coated with an example graphene, reference electrode silver chloride, and counter electrode carbon, general carbon. So this is how variety of immunoassays are being employed for detection of various infectious disease. In the same manner, if we modify IgM, immunoglobulin M and immunoglobulin G associated to COVID-19, then we can tell one person that whether they had antibody in the recent course of time with asymptomatic or symptomatic. So the content of the today's talk is what are the parameters we were missing during this lockdown situation. We know researchers are working on various diagnostic approaches, and government is bringing various policies to cut down the import of toxic materials from one location to other locations. But still, there are several other dangerous things exist in our healthcare system. So this COVID is very recent issue to our environmental. But our own metabolic system has plenty of other very serious issues which we were missing. We were missed to look. So what would be the deteriorations or lethality from that? So that is the key point I wanted to discuss here. This is also another sensor material named black phosphorus, which is actually used some aptamers and antibody conjugated form as the sensor component, and tested the existence of antigens in the biological fluid. So apparently we not only from CSIR Central Electrochemical Research Institute, other part of R&D institutions in the country also vigorously working on various point of care testings. One is electrochemical approach, and another one is optical based biosensors for detecting the antigens or metabolic syndromes, by which we wish to provide a better health care to the individual as well as to the environment. So FELUDA, paper strip test for coronavirus, is the one among the technology developed from CSIR family from IGIB. The name FELUDA was coined after in the introduction of the specific CRISPR-Cas9 technology. So the F is denoted for FNSC 59 peptide sequence, by which a specific gene editing system has been modified on paper. Following the same pregnancy strip mechanism, researchers from IGIP in association with the Tata institutions, they have developed a paper strip-based COVID-19 sensing system, and which is also approved by Drug Control General of India for commercial applications. So this is the indigenous technology employed for detecting the coronavirus. So there are plenty of other antibody testing kits available in the market. One such famous kit is from Eagle Bioscience and Kuso Bio from China, and some of the lateral flow assays similar to pregnancy test kit for convalescent plasma samples were also recently published in research journals, stating that researchers are continuously bringing novel techniques for diagnosing the SARS-CoV-2 antibodies or antigens, or amplifying the RNA associated to SARS-CoV-2. Is my voice screen everything is fine? Can someone respond me?
Yes, sir.
Yes, sir.
Yes, sir.
Thank you so much. So
Yes,
in this slide, you can see
Screen is not visible.
yes, not visible.
Yes.
Yes, sir.
Visible.
Visible, sir.
Visible, sir. Visible.
For those, okay, thanks. For those it is not visible, I suggest to
Visible and
refresh your
audible.
screen so that it will be visible.
It is visible and audible.
Okay, thank you. So ELIXIS anti-SARS-CoV-2 is an immunoassay product from Roche Diagnostics, which actually using a electrochemiluminescence immunoassay methodology for detecting the antibodies of SARS-CoV-2. Recently, one of our institutions, CSIR-IGIB, IGIB, have started collecting plasma samples to screen whether we have asymptomatic kind of SARS-CoV-2 from our own CSIR employees. So using this kind of commercial available techniques. So the antibodies freely circulating in your blood samples will be monitored to tell how many of us have herd immunity. I mean, like one person in my working conditions has viral load just 2 months before, and I have a communication or contact with him in the past 2 weeks. But my immune system is very strong. I don't have any kind of immune response, meaning I don't have any throat pain, fever, cold or flu-like symptoms, but still my blood is having antibodies related to SARS-CoV-2. Then it is called as asymptomatic. So that kind of assay is performed by our own CSIR institution, just to tell the world community that how many of us are self-immune built against the pandemic situation and how many of us are symptomatic, meaning whether we were able to defend the virus without symptoms or with the symptoms. That distinctions people wanted to determine. So using these commercial testing kits, one thing we learned, what is the technology, whatever we do in R&D system need to fulfill the commercial need, meaning we have to make our own biorecognition element without the import supply from abroad, so that we can make a sustained technology for betterment of the communal health care. So you have the responsibility that being a teacher of prestigious organizations, you have to nurture this to young people, that we have to make our own technology to bring the fulfill product. So we know how to make biosensor platform, We know how to make prototype system, but we are not able to connect all these three people into one platform. That is what missing in present situations. So chemists have to do the biology. If you remember the Nobel Prize which was awarded recently for chemistry, those two ladies, Dr. Donna and another person from France, actually they are basically chemists, but they worked on biology. So interdisciplinary nature of research is very much needed of the present day. So without knowing physics, better chemistry or better biology will not come. So we are in a situation to learn interdisciplinary stuff so that a better inventions can be come. One example for that is this pandemic situation. Coming to the subject point of the present talk, what are the overlooked situations? So whenever this pandemic situation, lockdown is slowly relaxed, we were in a situation to go job, and we were in freely situation to roam in market for buying cheaper groceries, or we want to just meet our own friends to make gossips. So what are the factors deciding us to commute the safety from the virus? Whether my friend is healthy, whether the place I wish to visit is clean, whether I washed my hand before and after meeting someone. That is the main factor. And affordability, I mean, without taking much loss, whether my mission is accomplished, that is also an important factor. Because if we want to control our own affordability, then we may not be able to decide to go for a crowded place. Because how crowd is created? The crowd is created because of less demand and low price. So whenever we want to have our affordability at supermarket, we will go there supermarket. If not, we will go to some local market where many people will come and make contagious environment. So safety from the virus, again interlinked with affordability, eco-friendliness, ease of use, and time taken, comfortability. So these are the factors deciding us to commutate from one person to other, or one place to other place. So this is an important factor, how the transformation of contaminants from one location to other location is happened. So this pandemic situation has created several kind of changes in way of working, labor force, digital commerce, supply chain and manufacturing, continuity and resilience, environment and climate change, debt burden, and deglobalization. I don't want to list all the parameters. I think many of you are very familiar with some of this terminology. But one thing I want to recollect here is many of us are learned to use modern gadgets. We don't know how to operate a Zoom, or we never heard of this webinar kind of systems before. Until otherwise we face this COVID situation, many of us might just see Facebook and Twitter, nothing else. But we know whereby Zoom meeting, Google Meet, Google Form, whatever way of creating memes, seeing online courses, how to share the screen, how to capture the screen, how to type. So actually digital commerce is increased a lot during this pandemic situation. And supply chain and manufacturing, a shopkeeper learned how to reach his own customer despite of the situation happened in particular situation region. And development of new approaches to overcome the issues exist in particular sectors. Environment and climate change, so investor policies has been changed because of this situation, and some people have lost their jobs, which ultimately lead to poor savings or repayment of loans. Though government have taken serious steps to bring back the economy of individuals, but still there are instances of unemployment causes big psychosociological imbalance across the people. And deglobalization, previously we were importing product from China, product from Germany, or any other European countries through Amazon or Flipkart and so on so forth. But now deglobalization is actually running us. Even though Great India Festivals is going to start from Amazon or Flipkart, we are still worried whether the box the person carried to our home will have a clean situation or some contaminants. So definitely the purchase through online declined. We have to agree with that. So these are the variety of situations created in lockdown or pandemic situation that also has healthcare, that I will see in the next few things. So during this COVID-19, the number of death is actually confusing, because in our own location, I know one of my friend is residing in an apartment. The security guard of that apartment is aged about 74. Unfortunately, his son was passed away because of this COVID-19. He was at the age of 24. He was working in Coimbatore market in Chennai. Because of COVID-19, his life is lost. But when his father visited the funeral ceremony and everything in Chennai, he also had symptomatic COVID-19, but he survived. So it indicates something else. COVID-19 alone not killing the person. This is my own opinion. Please don't take this as a scientific evidence or something. Whatever the content hereafter I'm going to share is my own opinion based on my understanding from the literature available in the scientific journals or newspapers collected from various content. So it is a disclaimer statement. Whether to take or not take, it is all up to your individual perceptions. So comorbidity is the main factor actually determining the death of individuals who suffered from COVID-19. What is meant by comorbidity? It is defined as the co-occurrence of more than one disorder in same individual. For example, I have a stomach pain. I want to see a doctor. For example, GI tract, gastrointestinal tract doctor, because I have a stomach pain for a longer time. When I visited the hospital, I meet other person who is having cancer. Unfortunately, a cancer patient, stage 3 or stage 4. And also I see other person is having cough, serious cough, maybe because of asthma or other chronic obstructive pulmonary disorders. So when I see these patients, if my own family member is sitting there, my perception will be different. If I see my neighbor or my other street person, my perception will be different. I may get that infection. I may get any contagious touches from those areas. So that psychological behavior will create other disorders in your healthcare system. This may be acute, but when you return back to home and if you start a feeling of cold or flu, then your illness will start to keep on rise, and it creates stress markers that eventually lead to mental disorder. So whenever someone is having depression because of heavy workaholic nature, their stress level will be very, very high, and they may be having ulcer or other symptoms, acute ulcer. If it is not properly treated, it will lead to chronic ulcer. If chronic ulcer is not treated, it will create cancer. So comorbidity is the serious health concern actually determining whether your immune system will survive this pandemic situation or not. So this is the key point we have to think whenever a person is having COVID-19 or death happened in COVID-19. If we understand this thing, then our healthcare system can be protected in better way. I'm telling that based on these statistical information. You see here, this is the fatal figures published in late 2016. In 1990, we have serious death informations because of diarrheal disease, lower respiratory infection, preterm birth complications, ischemic heart disease, and tuberculosis. In 2016, you can see number 4 is shifted to number 1. Ischemic heart disease is coming into a first player. Second one is diabetes. In 1990s, nobody is having diabetes because we never heard pizza or burger or any other junk foods. So diabetes came into play in 2016 or maybe late 2010. Self-harm, maybe because of repeated use of intoxicants. COPD, chronic obstructive pulmonary disorders. In a street, At those times, maybe only one motorcycle or maybe one car in particular region. But right now in a family, son is having Pulsar, father is having Activa or something, and mother is having some other motorcycle or car. So every individuals have several amount of access to automobiles, modern gadgets, which apparently change our lifestyle. So lifestyle associated diseases are linked with ischemic heart disease and other metabolic diseases. You see high BP nowhere in the 1990s. Or people may not visited hospital to tell that I'm having something, whether giddiness or headache. People just used to live with high BP or diabetes. Now because of media and access to hospitals, private hospitals and so on, we came to know that several diseases exist in our environment. So the content actually I derived for this present talk is based on the book written by William James. The art of being wise is the art of knowing what to overlook. So if we learn what are all the things we have to worry or what are all the things we don't have to worry, then our health problem will be solved. So that is the key extract of this book, The Principles of Psychology. So the chapter 10 says the consciousness of self. So if you assess your constituents or whatever the constituent in and around you, then the feelings and the emotions can be controlled according to that. If the actions are required to face those emotions are controlled, then your systems will be preserved. I want to take this proverb or message in line with the COVID-19 management or any other future pandemics which is about to come in near future. So understanding our constituents and preparing our self feelings or emotions to face them and prepare the actions accordingly will preserve ourselves from endemic or epidemic or pandemic situations. Because infection is not alone our environmental problem. We have plenty of other healthcare issues, because every city has now multi-speciality hospitals having cardiology center, chest medicine, critical care, dental, dermatology, diabetology, ENT, general surgery, gynecology, hematology, nephrology, and so on. Neurosurgery, orthopedic, pediatric, oncology, neurology, rheumatology, urology. So there are plenty of multi-speciality hospital waiting for us. If we don't understand our own healthcare system, then we have to look at any of these multi-speciality hospitals, and we have to pay a lot. Not necessarily the money, our mind, peacefulness. So how to face those things, how to control our feelings, or how to prepare our action for fighting against the infections or metabolic disease. So we have our own system, that is host, and we have our own soldiers within our system, that is called microbiota. I hope many of the biology teachers are very familiar about the term microbiota. So that is the biggest determining factor of our healthcare. So individual microbiome, taxonomic profile, and metabolic capacities may affect the balance between pro and anti-inflammatory responses, between the suppression or enhancement of virus or any other pathogenic conditions. So if we maintain good microbiota in our host system, we are already ready to defend bacteria or virus or any other pathogenic contents. So the term microbiome describes the genome of all the microorganisms. It may be symbiotic, it may be pathogenic, living in, on, and all vertebrates. We have all the microorganisms, optimistic good bacteria and bad bacteria in our biological system. Whenever there is a situation arise, everything will get into play. The gut microbiome is comprised of collective genome of microbes inhabiting the gut, including bacteria, archaea, viruses, and fungi. So what they do play in protein synthesis, lipids, and polysaccharide functions. So basically their environmental conditions is our host system. So they control the signaling molecules, toxins and other organic metabolites required for our own normal functions. So in short, a reasonably well-defined habitat which has distinct biophysico-chemical properties. So these are the functions of microbiome. So weight of human organs. So there are different organs in our biological system. As displayed here, every organs are having different amount of weight. Can anyone tell which is having the highest weight?
It's liver, sir.
Liver.
Thank you. Thank you. Liver is the largest organ as displayed here, but the microbiome is the largest organ. You may have never heard of as an organ because that is the microbiome. Even though it is not our organ, but that is actually weighed about approximately 3 kg. So our entire system, this 3 kilogram of microorganism is determining our healthcare system. For example, it creates the genetic informations, or it controls or mediates the protein synthesis. For example, in nose, mucus production is there, antimicrobial chemicals is there. Whenever we have running nose, at the first day we will sneeze, then running nose start, then running nose converted into juicy kind of mucus, then it become very solid, and when you cough, that sputum will be spitted out. So that is one particular example of microorganisms, actually not necessarily microorganism, that controls our immune response. So that's why when you have yogurt, curd or buttermilk every day, the microbiome populations can be maintained well. Same way, microbiome has different role in different organs. So there is a small video I'm going to play. Why is the gut microbiome important?
Sir, your voice is not audible.
Sir, your voice is not audible, please. Sir.
Actually, I'm playing the video. So
That's
whether
audible now.
you are, I didn't speak actually. I just played the video, just to see the video how gut microbiome is.
Sir. Okay.
I can explain. Yeah, please. Yeah. Yes, yes. Just a 2-minute video, just to see how microbiome help our immune system. Later on, I will continue my presentation. So this point is very important. We have Helicobacter pylori in our stomach. So whenever Helicobacter pylori population is higher, this happens because if we don't have good amount of bacteria, I think many of our Indian ancestors used to have a habit of doing fasting. Whether you spiritually believe or not, fasting is good, because the Nobel Prize also gave for autophagy. Autophagy means, what is meant by autophagy? Can someone help?
Sir, autophagy means just you have to keep a gap of 14 to 15 hours to your stomach.
Yeah, actually, it is the process by which kind of providing nutritions to your own system within our biological system. I mean, we are keeping intermittent fasting. Cells die. Research found that when you do fasting, cancer cells will start dying. So whenever you do fasting, the amount of microbiome in our own biological system will start fighting each other, by which the good bacteria will get higher amount of number. So if we don't have good bacteria in higher amount, the infectious pathogenic Helicobacter pylori as displayed here will get increased population, which ultimately lead you ulcer. I know many people who have regular meals but still have ulcer issues, because they have high amount of Helicobacter pylori in their stomach, which cause serious ulcer conditions. So here also I wanted to mention, so whenever you have good amount of microbiome in our gut, your neurological impulse is also performing good. There is a famous movie in Tamil Nadu named Vasul Raja MBBS. I think many Tamilians might remember the famous dialogue. So one scene used to come. Actually, a comedian used to mention that whenever curd is mixed with sugar, your brain will work very good, because when you add sugar and salt in curd, it gives suitable amount of medium for microbes exist in the curd. So the combination of salt and sugar will give good amount of medium for microorganisms in the curd. Whenever you take curd or buttermilk, it gives lots amount of microorganism to your biological system. So during the fasting time, if you take curd or buttermilk, it enrich your gut microbiome, that apparently help your neurological function as well as immune system. So another important factor we actually overlooked during this COVID-19 is nutraceuticals. So this situation must came because of poor physical activity or nutritious food and non-maintenance of microbiome. That is the key point I wanted to emphasize. So when you do regular physical activities, your immune systems are performing well. There was a study from Switzerland saying that people who involved in physical activities, they have collected the blood samples from them, and they all other pair of people, they have collected blood sample who doesn't do any physical activities. They separated the plasma from the whole blood and treated the plasma to cancer cells. So the cancer cells are not able to survive in the plasma which had actually the physical activity. So that study actually confirmed that when you do lots of physical activities, the proteins, defending proteins or healing proteins against the stress is actually overexpressed. So the normal cells and cancer cells, the level of proteins is different. Healing proteins level is good in normal cells, those who undergo physical activities. Those who don't do physical activities may not have healing proteins, but will be vulnerable for infectious attack. So we have to maintain regular physical activities as well as good nutritional diet, by which you can maintain antioxidant and other things. So there are plenty of endogenous and exogenous antioxidants as listed here: glutathione, thioredoxin, ubiquinone, uric acid, lipoic acid, bilirubin, which is non-enzymatic antioxidant, and enzymatic catalase, superoxide dismutase, glutathione peroxidase. So these are the list of antioxidants internally exist in our system, resulted from metabolic process. And these are the exogenous antioxidants, maybe come through dietary sources, which are found to be the key for maintaining the scavenging of reactive oxygen species exist in our system. So eternal health is the main thing we missed during this COVID-19 situation. Under the eternal health, what are the determining factors? That is microbiome, antioxidants, mental health, psychosocial behavior. These are the main factors we actually forgot to maintain. If we continue to maintain these things, then we don't have to worry about this COVID-19. Not necessarily this COVID-19, any other future endemic or pandemic situations. So this is all can be influenced by mainly social impact, like isolation, quarantine, which can actually create sleep pattern changes in sleep pattern, and lack of knowledge on infectious disease. If we have any situations, we have to first try to understand the conditions, how the infection pathology happens. If we understand that, then we can actually be conscious at wherever required and wherever not required. And we have to learn the adaptability skill, so that the changes, whatever happening in and around us, we can fight against that. And lack of physical and mental knowledge, that is also very important, which can be maintained through physical activities. And impact on economy, if we really worried about the economical situation growing up and down, that will create anxiety and depression, stress, that also can hamper your mental health. And fear and anxiety on individual health. Some people very healthy, but still they feel that they will have any infections when they go out or contact with the person who recently traveled somewhere. So if we understand these eternal health, constituting microbiome, nutrition, diet-rich food, and maintain our mental health through physical activities, and maintain good psychosocial behavior, obviously we can overcome any other situations arising in the future. That is the content I wanted to convey in this presentation. And there are plenty of research in line with the diagnosis is happening in our institution, in addition to other prestigious laboratory across the country. And my own research group is working on electrochemical immunosensor for making viral immunoassays related to dengue. And also we have recently started working on COVID-19 spike protein diagnosis in association with ICMR institution in Chennai and other private institution in Dr. MGR Educational Institute in Chennai, funded by DBT. And we try to come up with interdisciplinary approach, integrating chemical, biological and physical knowledge, people or expertise, to bring our own sustainable indigenous technology for healthcare. Finally, I wish to thank our head of the institution for permitting Jigyasa coordinator and recommending myself to discuss something about the current situations. I wish to record my sincere thanks to Jigyasa coordinator for giving me an opportunity, in addition to my divisional head Dr. James Joseph sir and director madam Dr. N. Kalaiselvi. I especially thank my PhD scholars and project assistant working on my ongoing project. Finally, I wish to also express my sincere thanks to members of EEC and staffs of Central Instrumentation Facility CSIR-CECRI for supporting our ongoing work. Thank you. That's all for today. If there is any questions, I will be very happy to answer.
Good evening, sir.
Thank you. Yeah, please tell me.
Therapy, please explain again. Plasma therapy, how?
Yeah, plasma therapy is nothing but, I told you now, plasma will have the antibodies. So if a patient is already recovered from COVID-19, they will be having antibody. Imagine polio virus vaccines. Polio virus is a attenuated vaccine, right? So your baby or any other family members, if have polio virus vaccine, our immune system will generate copies of antibodies for that antigens. So whenever in future we have the pathogens coming from environment, our memory cells having the memory of generating the antibody to attack those uh, antigens will started synthesizing. So the COVID-19 recovered patients will have antibodies in their circulating blood. So whenever they donate plasma, the plasma is identified to have antibodies, which also called as neutralizing antibodies for the antigens, that is SARS-CoV-2. So if they donate and your blood also has no incompatibility, I mean like immune response. I don't know whether, of course, I'm not a core biologist, but the plasma therapy is, this is how it works. The plasma will have neutralizing antibody. So when you have small amount of neutralizing antibody in your immune system, our cells will start to generate or make a Xerox copy of those antibodies by which you can collectively attack the antigens or viral particle coming out from environment. But remember, any flu vaccine, the validity or expiry will be 1 year. So another year you need a booster dose. In Canada and other cold countries, every year the flu vaccines will be injected. So I also believe the same for COVID-19. Even though if vaccine comes to in market, it need booster dose or regular dose, because you see from 1997 the SARS H1N1 H5N1 is coming in various forms. So they will be having continuous mutations. So we have to repeatedly take the booster dose. I hope I answered your question.
Sir.
Sir, good evening,
my
sir.
question. Good evening, sir.
Yes,
Good,
yes.
good evening, sir.
Yes, please.
Sir, I have a question.
Yeah.
Sir, is it true that first vaccine will be invented, then virus will be created?
That's a $1 million question. Nobody knows. Actually, it somehow, coronavirus is not new. It is exist in very old. Right now, we have technology actually to screen the electron microscope image of SARS-CoV-19. But in my slide itself, I mentioned, right?
Yes,
In
sir.
one of the slide, I mentioned electron microscope of H5N1 and other influ viruses. I mean,
Yes,
like
sir.
in 1997 itself, coronaviruses of sister family has been identified. It not necessarily engineered or purposefully spreaded. I think it exists in environment and started mutating and become living.
Yeah, sir.
Thank you,
That
sir.
is my question, sir. Whether global warming has given some impetus to the change or mutation of these kind of virus, starting from H5N1 to now we are taking H1N1, swine flu, and now COVID-19, or it has decreased our immune system.
It's either way. Say the resistance development will start at both side. Whenever you have penicillin antibodies in olden days, right now we have tetracycline, ofloxacin, or other ciprofloxacin kind of antibiotics. The resistant development can happen at both end. Sometimes virus or bacteria will start at mutation, and sometimes our own immune system will start respond against differently. Right now, people also think that Maharashtra, Gujarat, and other northern part of India has some virulent virus, but South India has kind of different strains. It is India is a very big country, and mapping them individual mutant variants will be time consuming. Without studying those things, it may not be easy to come to a conclusion. So
Yes, sir.
I see some couple of questions in the chat box. One of my relatives underwent antibody test for COVID. Which one is used, paper biosensor or something else? In hospital, ELISA based technique might be used. So basically, optical based method, paper based sensor, if they have given sample like oropharyngeal or nasal pharyngeal, they might be used RT-PCR. If they have collected blood sample, they might have separated the plasma and checked the antibody using ELISA or ECLISA techniques. I hope I answered some of the mentioned question. And temperature has direct impact on coronavirus. Yes, it is true. So surfactant can break the cell membrane of virus particle, and temperature also a key player by which the moisture exists our environment will denature the virus particle. That is why the lockdown was introduced. So whenever 36 hours or 45 hours is the virus is laying in the environment, if doesn't have any host to sit and multiply or hijack the system, then they themselves will die or denature in the environment. So to answer your question, temperature, yes, it has a role. How much possibility is there would be lab born? I really don't know. I have to go China and secretly talk with someone to tell this answer. I'm sorry to tell that lab born. I don't know that. Any other question?
Excuse me, sir.
Yes.
Sir, we are trying for finding the vaccine for the virus.
Yeah.
Do we have any antibodies to kill that? Because there is no prevention we are doing. But once it occurred, can you kill that virus in the biological system itself? Because we don't have any antibiotics. We are trying for that.
That is how people also worked about this convalescent plasma treatment. Plasma treatment is nothing but antibody, neutralizing antibody treatment only. But one thing we have to see, if these neutralizing antibodies give any adverse effect, that may be very serious, right? So vaccine is better than antibody treatment, because vaccines will have the purified specific fragments of viral antigens. So once it comes to our biological system, our own body will engineer or cook food for our own self. But when we take antibody, you are eating the food which was cooked by someone else for someone's purpose.
But so purpose, we are telling that alcohol and detergents the proteins can be dismantled, the virus protein. Alcohol works as a, so why can't they be used for the treatment itself? So what actually antibody the virus, once a virus enter in our body, why can't this alcohol work for killing the virus which is in our body? It can kill in the hand, why can't it kill in the body?
Sir, the antibody therapy it's a practice. Even in cancer, monoclonal antibody therapy is in practice. But the problem with the COVID-19 would be personalized screening is nobody have done. See, if I get admission with antibodies given or donated by somewhere, I don't know the adverse effect. Clinical trial may not happen for those antibodies. Right now, Bharat Biotech and many other pharmaceutical company approved by ICMR and WHO guidelines, people are screening various amount of vaccine leads. So which need phase 1, phase 2, phase 3. So without doing all these clinical trials, we cannot come to a conclusion that antibodies can be directly used or not. Because cancer antibody therapy, though it is well tested in various models, still not completely explored. So there are plenty of study required.
Okay, thank you, sir.
Thank you. Let's see how to make vegetables safe to touch when we bring it from marketing. Just make it wash with water or with salt. That is more than enough. 5 minutes, 10 minutes soaking, that is more than enough. I don't know whether it is good or not. Whenever I go to ATM and I use the touch panels, right after the touching of the pads, I just to rub my hand. See, that mechanical rubbing can also sometimes generate heat, right? So if I have moisture of something, contaminants, I just want to generate heat locally so that I don't know whether it is really good or not. But yesterday also I went to ATM machine to withdraw some cash. Right after touching the pad, I just wiped my hand. I didn't have a hand sanitizer on my packet, but I just make a heated rub, so by which I generate some heat and remove my moistures, whichever contaminated from some sources. So washing, I think it's well advertised in the television. Washing with a simple salt is more than enough. So to answer the nature, it is our own system, because Ebola virus, this SARS-CoV came from other mammals like bat. Those doesn't have ACE2 receptors, angiotensin converting enzyme receptors, so they don't have defending mechanisms for SARS-CoV-2 virus. But ourselves, we have ACE2 enzymes for our physiological functions. Because of that, we are providing sitting space for viral antigens, and we develop immune response. So we can't control that. It is the theory of evolution. Yeah, I mentioned that to answer Nirmala madam, I mentioned that mutations will keep on go on. That's why every year you have to have small amount of vaccines. We have to regain our memories of vaccines. I hope I answered all the questions posted in the chat box. Whether COVID test is compulsory for all, and if it is so, which test is preferred? So it depends on the locality what kind of question is test is practiced. So if they want to tell whether the person has had infection in the past, then they can go for antibody testing. As I mentioned, ELISA or electrochemiluminescence based antibody testing can be performed. If they don't have previous travel history, but they are if they have previous travel history and want to tell whether they are good enough to travel somewhere, then RT-PCR can be a better analysis. We were wearing from 90 since past 3 months here. The COVID thrice report is so other. See, there are plenty of viral infections. Sometimes anxiety. Some people normally having a body temperature of 101. So if they are able to eat properly, sleep properly, and do the natural activities fine, means no need to worry. I think I'm not a core clinician to answer some of the symptoms related question. But still, there are other virus infections may also can cause you continuous fever. Thank you. There are few cases where people had second COVID infection. Will the vaccine that is developed really work? See, that is what I also told. I was also told pneumonia can cause by both bacteria and virus. So in recent days, pneumococcal vaccines is available for babies less than 5 years. It does not mean that you will have pneumonia infection in future. If pneumonia is caused by bacteria, that will be prevented. If pneumonia caused by virus, that cannot be prevented. So you have to have personalized vaccines or personalized medicine so that you will be recovered from COVID infections. So your friend might had, or some of the cases might had some other infections which may be wrongly diagnosed as COVID-19. Then because of their previous medications, their immune system might be disturbed. So whenever antibiotic is preferred, you have to consume the antibiotics with vitamins. After the infections, you have to have good amount of nutritious food so that concurrent infections will not come. So again, if COVID infection or any other infection is coming, means your immune system is slightly disturbed or suppressed because of your previous health conditions. So nobody can assure continuous recovery. Any other question?
So this 99 to 100 degree temperature in present scenario, is it normal?
That is what I mentioned. I mean, like if they feel comfortable, I mean, like if they are able to eat properly, drink properly, they don't have any other symptoms, throat pain, something, then you don't have to worry. See, you never know whether the thermometer is worked fine. You have to check with some other thermometer or some other way of diagnosis. Whether you checked the same with the same thermometer, or you tried also with a different thermometer and still found the same temperature fluctuations.
Yeah, tried, sir, tried.
But still 9900.
Yeah, varying.
If there is no body ache, if there is no sneezing, cold, flu kind of symptoms, I think you don't have to worry, because sometimes some exceptional people will have normal temperature of 101. That is what I feel personally, because one of my own colleague had actually some kind of flu symptoms, but he tested 2 times negative for COVID-19. He had some ulcer issues. That ulcer actually made him some kind of aggravations and caused repeated fever kind of symptoms. So when they reached normal physical activities and other social behaviors, the temperature gone down.
கபசுர குடிநீர் வந்து எப்போ வேணா எனி கபசுர குடிநீர் கவர்மெண்ட் இஸ் கிவிங் how many times you have to take and respirometer can we use?
I think kavasura kudineer 3 times or 4 times in a week is okay. See, that has kavasura kudineer also has high amount of nilavembu. That is the major constituent. So whatever the bitter constituent in herbal formulations, better to take 3 to 4 days. That is more than enough.
In a week.
Yeah, in a week. Or better.
Good evening, sir. Good evening, sir. My question is.
Your mic is muted, sir.
Respirometer we can use, huh? Respirometer.
Sorry, sir.
Respirometer. Respirometer.
I think you check the oxygen level.
Sir, after giving medicine for this COVID-19, what people are suffering is they are having blood clotting. So how come this COVID is helping them to means themselves to get in a proper physical posture and a proper physical activity, sir?
Sir, this blood clot symptoms is diagnosed in Indian patients. Actually, the receptor angiotensin converting enzyme I mentioned, spike protein of SARS-CoV-2 will bind to ACE2 enzymes. So it is actually a blood clotting factor. One kind of it has metabolic role in blood dilutions and things. So when virus particles is circulating in our bloodstream, our immune system will start attack them and try to engulf, kind of phagocytosis process. So when cytokine storm is happening, this term might be very familiar from the television displays. Cytokine inflammatory markers is repeatedly synthesized by our body. That is the reason for high temperature, body pain. So the repeated amount of release of inflammatory markers make the clot. If the person already had diabetic or hypertension or any other things, then there may be sudden drop of oxygen flow into particular organ, which creates organ failure and sudden death. I think this is the reason for that blood clot happened in COVID-19, but this is not for all. Some cases, because of their comorbidity. That is what I mentioned in the presentation. Comorbidity is the serious factor. Okay, if there is no other question, thank you so much for patient listening, and I would be happy if there is any other question. I can respond that by email through Jigyasa coordinator. Thank you for patient listening. All the best. Thank you all.
Thank you, sir.
Thank you, sir. Thank you, sir.
Thank you, sir.